Hey everyone, so today we're going to start chapter 10, Microbial Metabolism, part 1, the metabolism of microbes. This chapter is often a challenge for students because it involves a lot of biochemistry in action, because we're going to really study and understand about how enzymes work, um, and how metabolic pathways work, particularly catabolic pathways, the breakdown of glucose, ultimately to carbon dioxide, water, and boom, lots of energy. That's the whole point of it, is how do cells get their energy to carry out all the work that they have to do. So let's get started in this first section, the metabolism of microbes. Now, keep in mind also as we go through this that many of the topics will be the same for eukaryotic cells as well as prokaryotic cells, and we'll make notes where there's differences and what those differences are. So these are the learning outcomes for this particular section. So we're going to understand metabolism, catabolism, and anabolism. We'll talk about enzymes and the parts of enzymes, apoenzymes and haloenzymes, and we'll talk about when and where enzymes are made and whether they're released or not, and whether they're made all the time or just some of the time, uh, different patterns of metabolism, and how enzymes are controlled. So what does that word metabolism mean? So a lot of times people think metabolism is just the catabolic part of the equation, but metabolism is actually all of the chemical reactions and physical workings of the cell. So really everything the cell is doing, building new cell membranes, making cell walls, making enzymes, making toxins, making DNA, breaking down all of those things in order to generate ATP, using their flagellum, all of those things are associated with metabolism. And we can split that into two areas. The first one is anabolism or anabolic reactions. These are the biosynthesis or building reactions. So this word biosynthesis, bio life, synthesis to put together, to make something. So that's the building part, and that part is going to require energy. These are energy requiring steps. Whereas the catabolic piece is breaking the bonds and releasing energy. Now, what we'll see as we go through this is the anabolic reactions and catabolic reactions are often going together. So in other words, if I'm building something, I'm going to need energy in order to build it. How am I going to get that energy? I'm going to be breaking down something else in order to get the energy that I need to build that particular reaction. So this is kind of a nice overview of all of the biochemical reactions of a cell. So starting with our friend glucose. Remember glucose, C6H12O6, you've probably heard that before, and other nutrients from outside the cell or from internal pathways if they've gotten built up there. The first part of it is to break stuff down. And this is the glycolysis, the Krebs cycle, the respiratory chain, and fermentation. And this is actually what we're going to be spending all of our time on, most of our time on in today's chapter. We'll spend a little bit on the anabolic side, but the majority of our time is going to be spent on the catabolic side. So once you break stuff down, you end up with precursor molecules like pyruvate, acetyl-CoA, and glyceraldehyde 3P. And those building blocks can then be used to build amino acids, sugars, nucleotides, and fatty acids, the four major macromolecules of living things. And those will continue to build till we get proteins, peptidoglycan, RNAs and DNAs, complex lipids, build, and then finally we have an entire cell that we're building. So all of this is part of metabolism. So, in order to facilitate these reactions, we need to use catalysts. And catalysts are in biological systems are enzymes. And so enzymes are biological catalysts, and a catalyst is something that speeds up the rate of a chemical reaction without becoming part of it. So they facilitate that reaction. I kind of think of it like if it's a matchmaker, right? So if you have a matchmaker, and say I'm a matchmaker, and I see one person over here, and I think, oh, that person would be so good with this other person. So I go about ways to introduce those two people together. And then I step away because my work is done. That's what catalysts do, right? They put things together, or sometimes maybe the catalyst is the one that's going to go in and break them apart. Either way, they don't actually become part of the reaction, or in order they get consumed in the reaction. But in order for enzymes to do their work, they need to overcome the activation energy. And we'll look at a couple of those curves in just a moment. And to do this, um, they allow the reactions to proceed because they'll either increase the thermal energy, increasing the speed of the molecules. That's one way that we can overcome activation energy. We can increase the concentration of the molecule, so they're more likely to come in contact with each other, or we can add a catalyst. So these are three ways that we can overcome activation energy, but um, enzymes are a great way to do that. This is a checklist of what characteristics an enzyme has. I encourage you to take a little bit of time to go over this and make sure you understand what all the words mean, and make sure that you understand how this all fits together. So for our purposes, we're going to think of enzymes as proteins, and we'll see that they may require cofactors or coenzymes, but they're typically made out of proteins. And remember, proteins are amino acids. They act as organic catalysts to speed up the reactions in cellular reactions. They lower the activation energy. So that means that they enable the reaction to occur without needing to heat it up or increase the concentration, things like that. So they help facilitate a chemical reaction to proceed. They will have unique characteristics. So each enzyme is different. Each enzyme carries out its own reaction and is different. So you can think about all the various biochemical reactions that happen in a cell. Cells need to make a lot of enzymes. E. coli, for example, carries about 4,000 genes, and well over 1,000 of them are enzymes. So, there are other proteins that are involved, that things get made, structural proteins and that sort of thing, but the majority of proteins are enzymes. They enable reactions to um, be carried out at a speed that's compatible with life. And what that means is that many of these metabolic reactions would occur in nature, but if without enzymes, they would occur so slowly that cells wouldn't wouldn't be able to exist because they'd be taking so long to wait for various reactions to occur. They have an active site for targeting molecules that we call substrates. So the substrate is what's being targeted by the enzyme. And although they get closely associated, it actually will form temporary bonds with the substrate. They don't become integrated or part of the reaction products. So the enzyme might be temporarily modified or changed while it's going through carrying out its activity, but at the end, it goes back to where it started. Just said permanent, not permanently changed. They are greatly affected by temperature and pH. And this is why cells need to maintain those things. That's part of why cells need to maintain homeostasis. And they have feedback and genetic mechanisms to regulate their functions. And we'll talk about a lot of these things as we go through the talk today. So how do enzymes work? Well, we already mentioned the term substrate. This is what the enzyme acts on. They bind to the substrate and change it in some way. Okay, so let's watch this brief video on how enzymes work.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur. To understand how making it easier for the reaction to occur.
So I just wanted to pause it right here for a moment just to point out these curves. This is what we were talking about when we mentioned energy of activation. So it requires some energy input to get those molecules past that transition state and to flow into whatever the products are going to be. When you have an enzyme present, it actually lowers the energy input that's required and helps to stabilize those reactions.
To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell in every living creature on the planet.
So hopefully that gives you a better appreciation. I think that I like this video because it reminds us again what proteins look like. It reminds us of the structure of proteins, the shapes of the proteins, and then we actually see some of the molecules and atoms that are involved in these reactions. Obviously, you don't need to memorize those particular the details of that reaction, but I think it's important that you can kind of see how these enzymes are actually occurring. And even though it looked like it was kind of happening quickly in the video, it's nowhere near as fast as it actually occurs in real life. Um, these these cells can carry out these reactions. Enzymes can carry out reactions millions per second. It's just kind of mind-boggling when you think about it. So what is the structure of enzymes? Well, the structure is there for us, we're going to think of them as being made of proteins. And we just saw kind of how they look in that video, kind of like these globular proteins. They can be classified as simple or conjugated. A simple enzyme is just the enzyme itself, just the protein. But many enzymes require other things in order to be complete, and we call those conjugated. They can either have pro, um, cofactors or coenzymes. Coenzymes are organic molecules that need to be bound to the enzyme, and cofactors are inorganic metal ions that need to be bound. So things like magnesium, zinc, that sort of thing, and coenzymes are things like vitamins and that kind of thing that need to be bound to make an enzyme functional. A lot of times you hear people talking about, oh, I'm going to take zinc, or I'm going to take vitamin C, or I'm going to take both of those things to boost my immune system. And what they're hoping for is that the vitamin C and the zinc will actually help the enzymes that are needed by the immune system to function better, to help prevent you from getting a cold, boost up your immune system so that it's more able to respond. And those enzymes will be better able to respond if they have all of the proper coenzymes and cofactors available to them in order to carry out their function. So that's really why you're doing those things, or why those things might actually help. So when we have the cofactors and coenzymes bound to the enzyme, the protein portion of the enzyme, we call that a halo enzyme. When it's just the protein component, we call that the apoenzyme. Generally, I'm just going to refer to them as enzymes because that just is the easier way to term it. But when we say an enzyme, you should keep in mind that we're talking about the halo enzyme. We're talking about the protein portion, which is the apoenzyme, as well as any cofactors or coenzymes that are required. So just to remind us about proteins and protein structure, we have the primary structure, which is just the sequence of amino acids. And then they will form alpha helixes or beta pleated sheets and that sort of thing based on the chemistry of the amino acids that are the sequence that you have there. And those will further come together to form the tertiary structure. And notice an enzyme can have multiple active sites on one enzyme. If the enzyme requires more than one protein, then it would come together to form quaternary structure. And quaternary structure is just where multiple proteins are needed in order to form the final product. But again, involving all of the folding and structure that we see, and go back and look at that video and thinking about quaternary structures and things, and you'll see these structures, when you look at some of these videos. So just kind of think about that as you're watching these videos. Now, the active site, this is actually where the business happens. This is where the actual activity occurs, whether it's breaking down or building the reaction. And again, every enzyme has a different primary structure, a different sequence, and that's going to ultimately come from the DNA that's creating that sequence of amino acids. They're going to Based on that sequence, it's gonna change how they get folded, and that's also going to give them all a unique active site. And this is why we need to have so many enzymes in order to function. So I've already mentioned a little bit about cofactors and coenzymes, and so this is just to um, remind us of what these things are. So we can have metallic cofactors like the ions, iron, copper, magnesium, zinc, selenium, manganese. These are things that we need in trace amounts, if you remember back to the earlier chapter. And these assist with the functions of the enzyme. They help to activate the enzyme. They help to bring the the active site and the enzymes closer together. And they sometimes can participate directly in those chemical reactions that are occurring in the enzyme. Now, coenzymes are very similar in that except for they are organic molecules, and those are things like vitamins that will also bind to the apoenzyme to form out the haloenzyme. They often are important for carrying out and transferring hydrogen atoms and electrons, carbon dioxides, and amino groups. So they're often involved in carrying and transferring um, groups, functional groups, and yeah, so they're important parts of the cell. So again, these reactions happen extremely quickly, up to millions per second. And we just saw in that video kind of what that can look like with one example. And here's a static way to look at it, because sometimes we can't our minds can't go a million per second. At least not consciously. So for a reaction to take place, you have to have a temporary binding or action between the substrates and the enzyme. And we call that sort of a lock and key fit or an induced fit, where the enzyme will actually fold around and hold those substrates in place. The bonds that are formed there, however, are very weak and are easily reversed as the products get released out into the environment, whether that's the cytoplasm um, or wherever these enzymes are located. So what are these reactions that these enzymes are carrying out? Well, they're pretty much carrying out one of two reactions. And we'll see that there's always variations on these reactions, but these are the two primary reactions that are that are happening. And so let's take a look at those. So the one on the left is a building reaction where you're starting with two molecules, two glucose molecules. And if you remember, glucose molecules have these OH groups, and you put in some ATP, and here's our enzyme, and it's holding those two glucose molecules, and we're like looking inside it. And the energy from the ATP goes into forming this glycosidic bond, and it's the removal of a water, and we call this reaction a dehydration synthesis. And even in that short video we watched, he talked about dehydration. That's what he meant, the removal of a water. And every time we're building something, whether we're building sugars, here we've built a disaccharide, or whether we're building a lipid, or whether we're building a protein, or whether we're building an amino acid, or a nucleic acid for that matter, it's always a dehydration synthesis, the removal of the water. Now, the details of the bond will be very different. This is a glycosidic bond. Between amino acids, we form peptide bonds, we can see here, peptide bond here. Between, so you need to, form a phosphodiester bond. But even though the bonds themselves are a little bit different, the way they were built is always the same. They were always built by removing a water, removing a hydrogen from one of the molecules and a hydroxyl from the other to form a water. And that removal of the water then makes available a space for a new bond to form to complete those valence shells, if you remember your basic chemistry, and enable then that bond to form. Now, so that's dehydration synthesis on this side. This other side is a condensation reaction. So you'll hear it called condensation. You'll also hear it called a hydrolysis reaction. So what does that sound like? Hydrolysis, water lysis to break. So here, this is the catabolic side, the breaking down side of the reaction. And here we're putting water in, and that water is going to go into this reaction and it's going to break that bond. It's going to, in this case, it's two amino acids, so it's a peptide bond, and it's going to break that bond. And so it's going to, the hydrogen and the OH groups from the water are now going to bind to reform the carboxyl group in the one amino acid and the amino group in the other amino acid. And we end up with two amino acids there. You can kind of follow the water. This also will release energy. Energy releasing. Whereas we said the anabolic reaction, this side, I should write down anabolic. The building reaction required energy. So as long as you're clear on these two kinds of reactions, that will take you a long way to understanding all of the things that we're going to be talking about the rest of this chapter. So how do we classify enzymes? Well, we can classify them based on their function. We can classify them based on their site of action, the type of action that they carry out, and what their substrate is. So there's different ways of classification. And so obviously enzymes can be classified as more, in more than one area. So here are some examples of the six of the major classes of enzymes and what they generally do. So we have a group called the oxidoreductases. Oxidoreductases. These transfer electrons from one substrate to the other, and related, where'd my slide go? And related to that are the dehydrogenases that transfer a hydrogen from one compound to the other. And transferases, kind of what they sound like. They transfer functional groups from one substrate to another. Hydrolases are cleaving, and they're the ones that are breaking down molecules, breaking the bonds of molecules, and they're using water to do that. We just talked about that in the previous slide. Lyases add groups or remove groups to form or from double bonded substrates. So lyases are also adding or removing groups. Isomerases, do you remember what an isomer is? Remember, isomer is the same chemical formula, but a different structure. Isomerases are actually changing a substrate to its isomeric form. So it might be changing a galactose to a glucose or something like that. So making that switch in how those bonds are arranged is something that an isomerase would do. Ligases are kind of like molecular glue, and they form bonds using ATP, and of course, results in the removal of water when those reactions occur. So here's a sample of enzymes and their substrates and their reactions. You don't necessarily need to memorize these, but it's just kind of a good thing to just kind of go through and kind of say, okay, the common name for the enzyme is lactase or penicillinase or DNA polymerase or lactate dehydrogenase or oxidase. Notice they all end in ASE. And so when you see that, when you look at some of these big old long sciencey words and it ends in ASE, you should think, oh, enzyme. Then you're halfway there. Then it's just a matter of figuring out, okay, I know it's an enzyme because it ends in ASE. What is it doing? Well, let me look at the rest of the name of the enzyme. So penicillinase is going to break down penicillin. It's going to break the beta-lactam ring that makes up penicillin. We're going to talk more about penicillin structure in the next module, module 3. I guess it's module 3 that we do this one. But the enzyme class, it's hydrolase because it's a breaking down enzyme. Lactase is going to break down lactose. Okay. And so on and so forth down the line. You can kind of read these for yourselves and see what these are. But the big thing here is to recognize ASE as an enzyme. So transfer reactions are also carried out by enzyme. These are accomplished by oxidoreductases. And these carry out these redox reactions. We're going to talk a lot about redox reactions as part of glycolysis, the electron, the Krebs cycle, and the electron transport chain, because this is a big part of how cells acquire their energy are through these redox reactions and this transferring of electrons and hydrogens. So just to remind you, oxidation is a loss of electrons and hydrogens. And reduction is the gain of electrons, hydrogens. So if a compound is losing electrons, we say that it is oxidized. If a compound is gaining electrons, we say it's reduced. So just in the process of breaking down glucose all the way to CO2 plus water, which we're going to talk about all those reactions later, and this whole process is fully oxidizing glucose into CO2. So once glucose is completely oxidized, it's now in the form of CO2. And we got to figure out then, where did all those hydrogens, 6, H12, 6, where did all those hydrogens and electrons go? And we're going to follow that and figure out to the water ultimately. And we have these two coenzyme carriers, NAD and FAD. And when they are reduced, they are in the form of NADH and FADH2. So these are really important reactions to know. If you follow the electrons, as we go through all of these processes, just kind of keep in mind the electrons and hydrogens have to go somewhere. Where are they going to go? Um, Pay attention to the flow of the electrons. It's really what those processes are all about that we're going to be talking about in the next sections. So other enzymes that carry out transfer reactions are transferring functional groups like aminotransferases. They're transferring an amino acid, phosphotransferases, transferring phosphate groups, methyltransferases, transferring methyl groups, decarboxylases, removing the carboxyl groups. So, you can kind of tell the name, what they're doing by the name of the enzyme. That's kind of a nice thing. That they're named in such a way that we can sometimes tell what they are. We also can talk about them based on their location. Remember in chapter 9 when we talked about and learned how saprobes break down debris? And we said that they were making an exoenzyme. So exoenzymes are those that act extracellularly. They're transported out of the cell. They're made inside the cell because all the stuff that you need to make an enzyme, the ribosomes and all of that, that's inside the cell. But then they transfer it out of the cell. And so this is showing us, and then the enzyme can act on substrates that are outside of the cell. Very often that might be, a toxin or something like that, or it might be something that's going to break down nutrients like we saw with the saprobes condition situation, where they're breaking down large pieces of debris into smaller molecules that can then be absorbed and taken into the cell to serve as nutrients. Most enzymes are what we would call endoenzymes, and that means they just kind of stay inside the cell and carry out their work there. The enzymes of the metabolic pathways we're going to be talking about today, or in the future sections of this chapter, are all endoenzymes. We can talk about how enzymes are produced, the rate of their production. Are they made all the time? Are they needed all the time for some reason? If they are, then we would call that constitutive. So constitutive enzymes are made all the time, always on. And that means they're not regulated. There's no control systems to turn them on or off because they're needed all the time. They're made all the time. And this will be things where, it doesn't matter if you add more substrate, doesn't matter what is going on, they're just going to constantly being made at the same rate regardless. And those are very often going to be things like in human cells, we have beta actin, some of the structural proteins, the microfilaments, those kinds of things that are just constantly being needed by the cell. But most enzymes, or a big majority of them, and the ones that we're generally most interested in, are going to be called regulated enzymes. And these are enzymes that have an on switch, or sometimes they have an off switch. So if they're always on, they can be turned, but they can be turned, if they're usually off, is they're normal, but they have an on switch, we call those inducible. If they're normally on, but have an off switch, we call those repressible. And we'll talk a little bit more about those when we get to the molecular biology chapter, because this inducible and repressible component can also come in part to whether you make the nucleic acid, make the RNA at all to make the proteins. So sometimes these are switches that are turned on and off on the genetic level, but they can also be regulated on the protein level. And that's what we can see here in this graphic. So this one is showing that If we have some particular enzyme and we add more substrate, the enzyme is induced. So this one would be normally kind of in the off position, but when there's a reason to have it, it's going to be upregulated. You can turn it on. On the other hand, if you end up with an excess of substrate, you have more than what you need, we can have some feedback inhibition and that sort of thing, and that's going to turn this enzyme off or repress it. And we'll talk more about those as we go through. So what are the roles of these enzymes? Why do we care about them? Well, they're important in disease. Pathogens secrete their own unique enzymes, and that can help avoid host defects, that can promote multiplication. We call these things virulence factors. And virulence is described as the ability of an organism to infect a host and cause disease. And depending on what combination of virulence factors an organism has will kind of depend on how severe the disease is and and those kinds of things, and how likely it is to make you sick if you get exposed to it. All of these different kinds of things. Some of these enzymes that get secreted by enzymes function as toxins. A toxin is a poisonous substance that can harm harm its host. It's usually they can stimulate antibody production, so things like tetanus toxin and that sort of thing. We're able to make antibodies against to protect us, but not always, and not always fast enough if we haven't been exposed before. So here's a short sampling of some of the enzymes that different organisms can make. So streptokinase and digests clots and helps allow for invasion, and streptolysin damages blood cells and tissues. And it's also why we see beta hemolysis when we grow this on a blood agar plate. These are both made by Streptococcus pyogenes, the cause of strep throat. Elastase and collagenase are also made by Streptococcus oops, are made by Pseudomonas aeruginosa, and elastase is going to digest elastin, and collagenase is going to digest collagen. Those are both part of connective tissue, and so that's going to help with invasiveness if you break down the stuff that's holding cells together. Lipase is made by Clostridium perfringens. This is an organism that causes gangrene, and it's going to break down cell membranes, the lipids in cell membranes. And penicillinase is made by a lot of different kinds of organisms, especially if it's carried on a plasmid. It can be spread to other organisms, and it breaks down penicillin, specifically the beta-lactam ring that's in that class of drugs. We'll talk more about that in the next module, or in a couple modules. So we mentioned that enzymes are very sensitive to their environments. And remember in the last chapter, in chapter 3, we talked about the effects of temperature and pH on cells and cell growth. And a lot of the reason that cells are so sensitive to temperature and pH is because of the enzymes and the proteins that are needed to carry out that activity. So, you know, if the temperature gets too hot, or osmotic pressure changes, or pH changes, that can really affect enzymes. They can really only work in a narrow window or a narrow range. We talked about temperature. Remember, we had the minimum, max, and maximum temperatures that cells can live in and grow in, and a lot of times that's because the enzymes that they have need those particular temperatures. When enzymes are subjected to changes in their conditions, they can become very unstable, and we call those labile. And often enzymes can be heat labile, meaning that if you put heat into them, they will break down, become denatured, and no longer be able to function. So they'll lose they'll lose that tertiary or quaternary structure and just kind of fall apart. We see that if you cook eggs. When you fry an egg and the albumin in the egg that's normally that kind of clear jelly-like substance, that's intact albumin. When you fry the egg, it turns into that white cooked egg. That's the denatured protein. So metabolic pathways. Bacterial cell has more than 1,000 enzymes, and any all of these enzymes act together in these various pathways. Most of the time, various biochemical reactions are going to occur in pathways, meaning that it's not just one thing. It's not just one step to go from glucose to carbon dioxide. It's not just boom, one thing. It's a series of steps. And those pathways, in each step, is catalyzed by a unique enzyme. And in these pathways, the product from one reaction is often the reactant for the next reaction. And we will see that these pathways can have different sort of patterns to them. We'll look at that in just a moment. Some can be cyclic, some can be branching and different things. And these pathways are all highly, highly interconnected. So here are some general pathways. So we have a linear pathway, kind of how it sounds. A to B to C, A to B, B to C, C to D, etc. And an example of this we'll see in the coming sections is glycolysis. Glycolysis follows a sequential set of steps. There's cyclic pathways. An example of that is the Krebs cycle, where you have some inputs and it cycles in a continuous loop. And then we can have branch pathways, either divergent, where you start out in one pathway and then it splits into two or diverges into two. So an example for that would be protein catabolism. And then convergent pathways, where you have two um, two pathways that ultimately join together into one pathway. And an example of that is amino acid synthesis. And I just threw this slide in here to show you how interconnected all of these pathways are. This is someone who made a metabolic metro map, so it's meant to look like subways, how subways work. But if you notice, you can see, here's the citric acid cycle, here's glycolysis, and look at all the branches and arrows and all the different things. We're going to talk about beta oxidation and all of these different things to make fatty acids and lipids. All of these different pathways can merge and interact with each other in order to carry out all the metabolic processes of the cell. And just keep in mind, and this is actually an oversimplification, but keep in mind that every place you see an arrow is an enzymatic reaction. And notice some of these go in both directions, and that means that the enzymes can function in both directions. Sometimes it's just one direction, and the enzyme can only facilitate the activity in one direction. So we mentioned a little bit about control of enzymes. We talked about constitutive and regulated enzymes and how that is at the molecular level. And we'll talk more about that when we get to the molecular biology chapter. But when the enzymes have already been made, they can also have some kinds of regulation. And one is something what we call competitive inhibition. And in competitive inhibition, the inhibitor, and we can call it a competitive inhibitor, mimics the substrate. And that's really what this is saying. It mimics the substrate so much that it can come in and it can bind into the active site and block this enzyme from functioning. This is showing the normal way. So the normal way, the substrate comes in, it binds, and it produces the product. When you have a competitive inhibitor, it can come in, it can bind, and block action. Okay? This is important because this is really some of the ways we design drugs is to function as competitive inhibitors. So if you're overproducing something in your body, or if the bacteria is producing something and we don't want it to produce it, we can create a competitive inhibitor that will block that action, block that enzyme from being able to function. We can also have what we call non-competitive inhibition. And this is often what happens is the product itself will kind of come back and act as a feedback loop. So what does that mean? So here we have the substrate and the active site. And away from the active site, we have a site called a regulatory site. Sometimes you'll also see this called the allosteric site. Allosteric. And so what can happen is after you've made lots and lots and lots of product, you don't need to keep making anymore. You have enough. So when you have enough of that product, the product itself can come back and bind to this allosteric site, to this regulatory site on the enzyme. And when that happens, it causes a change. We call that a conformational change. So your enzyme is normally like this, and now the feedback has come and bound, and that's going to change the shape of the enzyme. And now the enzyme can no longer bind its substrate. Okay, so it's blocking further action by the molecule. Now, after some period of time goes by, if you start to run out of this product, this can actually fall back off again and you can go back to normal functioning. We call this feedback inhibition. We call this non-competitive inhibition. It's also called allosteric inhibition. It's kind of different ways of talking about the same thing. Let's watch this quick video.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
Okay, so This is a video that comes right out of your chapter. I don't usually or always use the chapter videos, but I do encourage you to watch them. I think a lot of them are very good and they're nice and short. Clearly, if you've already watched it, feel free to speed through this part of the
Many of the
talk. So controls of enzyme synthesis, we already mentioned a little bit about repression and induction. This is another way to regulate. This is regulating generally at the genetic level, so at the level of the DNA. So just to remind you, DNA, and we'll learn more about this later. DNA is the template that is used to make RNA, using a whole bunch of enzymes that we're going to talk about. And then RNA using ribosomes and transfer RNA and things like that end up making the amino acids and build, well, they don't make amino acids, they build the proteins. So repression happens that when the enzyme is normally on, it can be turned off. And we'll look at this in more detail when we get to those chapters. But if you kind of grasp it now, it's going to make that a lot easier then. So if you reach some excess of product, then it can bind to a repressor, and then that repressor will then come and bind on the DNA and block the production of RNA. And if you don't make the RNA, then you can't make the proteins. And of course, the protein that we're talking about here is an enzyme. Okay. On the other hand, induction is the opposite case. So again, we still have DNA to RNA to protein. But in an inducible, it's normally off, but can be turned on. And what happens here is, say it's for lactose or something that the cell isn't normally seeing. And so because the substrate isn't normally available, there's no reason to make the enzymes to break it down because you don't normally have the substrate available. But when the substrate becomes available, so normally in induction, there's normally a blocker, a repressor sitting on the DNA. But now that the substrate's there, it's going to pull that blocker off and make the DNA then available to start synthesizing the RNA. And so then it can induce the production of the RNA, which then makes the enzyme, which then can carry out the breakdown of that substrate. So the presence of having the substrate in the cell then will induce or cause this enzyme to be built and made. It's normally off, but can be turned on. And these are kind of opposites of each other. And again, we'll talk more about that in the molecular chapter, but it's really kind of important that you get to have a grasp on it in this chapter. So this is just kind of giving us a depiction of the genetic control. So here we have excess product binds to the DNA through a repressor and will block further expression and can't make that product. It's kind of taking us through this loop of DNA to RNA to make protein, and then the substrate then can come back and do feedback inhibition. Or do repression, I meant to say. So this takes us to the end of part 1 of this chapter. Take your time, go through, answer these questions. Again, follow the PowerPoint that's posted, because I do have some notes underneath the PowerPoint that help supplement the presentation, and have a good day.